everybody doing? Everybody's asking me about note cards. Where are the note cards? Note cards will be here on Friday. That means you'll be here on Friday. This is good. To remind you, and for those who haven't taken my class before, on the final exam, I let you have a note card. You have to get the note card from me. No other note cards are acceptable. And you will be required to have a note card at the exam. Okay? So if you don't have a note card at the exam, you're going to lose points, and you have to have that note card from me. So make sure you get a note card from me to use in the exam. You don't have to use the note card. Okay? I'm not going to make you use the note card, but you do have to have a note card for the exam. So make sure you get that, and I'll have those available in here on Friday. Okay? Okay, we're just about done talking about gene expression. I have one more topic that I want to address, and that is an interesting process in controlling both the translation and the stability of messenger RNAs in eukaryotic cells. Now, eukaryotic cells, as you've seen, have some complexity. There's a lot of complexity. I could probably teach 2 or 3 courses on eukaryotic gene expression. So there's a lot of complexity to ultimately determining what or the amount of protein that's made in a eukaryotic cell. So I want to show you one interesting example. You saw one interesting example in prokaryotes, which was attenuation. And I want to show you an interesting example in eukaryotes, which relates to iron. Iron turns out to be a limiting nutrient for cells in some cases, and in other cases it turns out to be something that is too abundant. So cells have to modulate the amount of iron that they take in and the amount of iron that they store. Okay? Iron left alone in a cell is a real problem. Okay, so if it's not bound up with something like hemoglobin or whatever, if it's just floating around freely in the cell, what iron will do is it'll create free radicals. And free radicals are giant problems for cells, reactive oxygen species, et cetera, et cetera. There are 2 proteins that I want to talk about, actually sort of 3, but 2 main proteins I want to talk about relative to iron inside of eukaryotic cells. The first of these is a storage protein, and it's called ferritin. Okay? Ferritin looks sort of like what you see on the screen. Colors added for effect. But what ferritin does is it gobbles up and grabs iron so it doesn't float freely inside of a cell. If a cell has too much iron, it wants to make more ferritin so it can gobble it up and store it so it's not floating around freely. Okay? If it has too little iron, there's no reason to keep making ferritin because the cell would simply be wasting energy. Okay? So ferritin is a protein that eukaryotic cells have to modulate according to the amount of iron that they have within them. Well, how does the iron get into a eukaryotic cell? It gets in as a result of action of a protein called transferrin. Okay? Transferrin is, interacts with a receptor called the transferrin receptor, and what that receptor does is it's basically allowing iron to enter a cell. Okay? So like we've seen other transporters that allow movement of other things into cells, the transferrin receptor allows the movement of iron into a eukaryotic cell. Now, we think about, well, what if the cell has a lot of iron? The cell has a lot of iron. It does not want to be making more iron, right? And if the cell making more receptor. And if the cell has little receptor, then it wants to make more so it can bring more iron in. Okay, so it's got 2 scenarios. All right. Well, it turns out they're sort of opposite, right? So if there's little iron, you want to make the receptor, and you don't want to make the ferritin. And if you have a lot of iron, you want to make the ferritin, but you don't want to make the receptor to bring more iron in. Right? So they're sort of cross purposes. All right. Well, the interesting way in which these proteins are modulated is as a result of another protein called the iron receptor element binding protein. Or iron response element binding protein. You can call it IRE-BP. Okay? The IRE-BP looks like this, and it's a protein, okay? And the IRE-BP can bind to two different things. It can bind to iron, or it can bind to a structure called the iron response element. So there's the confusion, all right? So I've got an iron response element binding protein. That's a protein. The iron response element is a structure. It's a sequence structure, as I will show you. Okay? So even though the names sound similar, they're completely different things. The P in the binding protein tells us it's a protein. IRE-BP is a protein. IRE by itself is a structure. Well, where does that structure come into play? The structure comes into play in the messenger RNA of the two proteins that I've described to you. The first protein is ferritin. This is what the messenger RNA of ferritin looks like. It has a stem loop structure. You guys have seen stem loops before. Here's a stem loop structure that does something different than the one you saw before. The one you saw before was involved in transcriptional termination. This one does not play a role in transcriptional termination. Instead, it's the target for binding by the IRE-BP. So the IRE-BP, when it sees this structure, will bind to it if the IRE-BP is not already bound to iron. You saw that it could bind to either iron or this structure in the last figure I showed you. Okay? If iron's present, the IRE-BP will bind to iron. If iron is absent, the IRE-BP will bind to this structure. When the IRE-BP is bound to this structure, translation will not occur, because the IRE-BP blocks the ribosome from doing anything. Okay? So what does that mean? It means that when iron is absent, okay, when iron is absent, the IRE-BP will bind to the IRE. Translation of ferritin will not occur, and that makes sense, because this is a situation of low iron. Low iron means IRE-BP binds, means translation does not occur, means ferritin is not made, and the cell is not wasting energy. That makes sense? Question? Yes? Okay, so that's the other situation. Her question is, what happens if iron is present or abundant? Well, if iron is abundant, then IRE-BP will bind to the iron. It will not bind to this structure, and translation will occur. Translation will occur because iron is abundant, and cell wants to make ferritin to bind the iron. Does it make sense? Okay, so two scenarios: low iron versus high iron. They lead to either translation or no translation of the of the ferritin protein. Yes, question. This is this structure right here is the messenger RNA for ferritin. Right? So the question is, will this messenger RNA be translated or not translated? When iron is abundant, this message will be translated. When iron is not abundant, this message will not be translated. Yes, Eric. What happens to all the ferritin with the iron bound to it? Well, it will release iron as it's needed in the cell. So it's mainly just a sequestration form for iron. Okay? Yes, back in the back, far back. If the iron is bound to the IRE-BP, is it available to make free radicals? In theory, there probably could be a little bit of free radical made wherever there's iron, but the fact these proteins are bound to them is really covering it up, and it's reducing that immensely. So the answer is basically no. Why doesn't the cell, so his question is a good question. Why doesn't the cell just make the IRE-BP? Because ferritin can bind many more ions. Ferritin binds a big clump of these guys. And so it's a more efficient way of holding on to iron that the cell needs. Yes, sir? Of this RNA? So this, okay, so the question is what regulates the transcription of the ferritin messenger RNA? All right? Well, there are things that will control that, but for our purposes, we can think of it as being made constantly. And the question isn't whether it's made or not, but whether it's translated or not. Right? So if it's translated, you're going to have protein. If it's not translated, you're not going to have protein. Okay? Yes?
Why cell sometimes chooses to have a control of gene expression, like on the level of messenger RNA, not before? Because it takes time to do the RNA.
So the question is why, basically, if I understand the question, basically the question is why does the cell have so many control mechanisms?
No, why doesn't it just limit itself to the controlling of the gene expression, like for transcription?
Yes.
Since it would be...
But that's the same question. So the question is, why doesn't it just control by transcription? So why does it have so many mechanisms? And the mechanisms allow cells to better sense the environment. In this case, it really wants to pay attention to how much iron is there, and this system gives it that. Okay? Now, it's always important to remember in these things, when we ask why, we're seeing the end products of evolution. We're not seeing, what brought that about in the first place. We're seeing the end products of a process that has evolved. And it evolved because it worked. Okay? So it's evolved because it's worked, and that working is functioning still today. So that's the best answer I can give for any why question with respect to gene expression. Yes? So his comment is that post-transcriptional regulation mechanisms allow a faster response to the environment. I would say in some cases, yes, they would. Yes. In this case, I think you can see that pretty readily. And this is half of the equation. We haven't seen the other half of it yet. Yes? Does this sort of regulation occur for more than just iron? That's a good question. I don't know of other ions in the cell that are regulated in this way. Cells have ways of dealing with, for example, toxic metals like lead and things like that. But that's a different mechanism than this. So I don't know of others. I'm not an expert in this, so there may be others that have a similar mechanism, but this is the only one I know that offhand that does that. Okay, so this is half of the equation. So we now know how ferritin is regulated. It's regulated by whether or not its messenger RNA is translated. And that messenger RNA being translated is a function of whether or not iron is present in the cell. Okay? Now, the other protein I told you about was the transferrin receptor. And I said it had an opposite issue with respect to being made or not in comparison to the presence of iron. If we look at the transferrin receptor messenger RNA, we see something really interesting. Okay? This is what it looks like. Now, you'll notice that it has 3 iron response elements in it. Okay? I'm sorry, 5. Well, I can't even count. It's got 3 up here and 2 down here. Okay, it's got 5 elements in it. All right? 5 elements. You think, oh, there's all kinds of places for iron response element binding protein to bind, and that's correct. However, look where they're located. They're in the 3' end, not the 5' end. I'd like somebody to speculate and tell me why or how you think this works. Or somebody who's watched my lecture and has already gotten the answer. Any thoughts? Yes?
Translation still happens, but however, depends on whether anything seated after this, it will be finished or not.
Whether it finishes properly or not? Is that what you're saying?
Well, yeah.
So the comment is whether translation finishes efficiently or not. No, that's not it. Okay? This one doesn't relate to translation. There's a hint.
This one doesn't work?
Doesn't relate to translation. Omar? It's no. But anyway, David.
So you said earlier that the IRE-BP binds to iron at a low iron level, right?
Right?
We want, when there are low iron levels, we want more transferrin receptor.
That's right.
All right, so the fact that it's on the 3' that must activate.
Okay, so David's on the right track. Okay, so he's made the observation that we want to be making transferrin when we have low iron. Right? So low iron is when when the IRE-BP is going to bind, right? Now, his question, it's a good idea. It's not the right answer, but his idea was that perhaps the binding of IRE-BP favors translation. Well, in a sense, maybe it does. I'll give you the answer, okay? The answer is that it's not controlled at the level of translation. It's controlled at the level of the messenger RNA stability. Remember that there are exonucleases present in cells, and the poly-A tail was a buffer against that degradation of the messenger RNA that happens for any messenger RNA that's out there. If you have on this messenger RNA at the 3' end, you have IRE-BP, what you do is you stop an exonuclease in its tracks. So this stabilizes the messenger RNA message when iron is low. Stabilizing it allows more protein to be made, and therefore more transferrin receptor is made to bring in more iron. When iron is high, IRE-BP binds to iron, not to the element, and this becomes unstable and is degraded during the normal degradation process. So it's a very cool way of having opposite effects on two proteins that have opposite roles in the cell. One present when there's high iron, the other present when there's low iron. Comments, questions? I see discussion. That's good.
Does the mRNA normally have a short poly-A tail?
Messenger RNA has a poly-A tail. I don't know if it's shorter than anything else. I don't know that it's shorter than normal. No. Yes? Why are there so many IRE-BPs? Okay. Well, it just gives more chance, more opportunity to block the exonuclease from going on. That's the only reason. Yes? Is it important for you to know what regulates the IRE-BP itself? Well, I haven't talked about it, and to be honest, I don't know, but we could speculate ourselves, couldn't we? What if IRE-BP regulated itself in some way? Could you imagine a scenario where it did? What would be the wants to regulate IRE-BP? Would you want it high iron, low iron? When would you want it?
Maybe there is a high iron rate just on level transcription. It would be binding to the operator region and just by this thing like.
Well, let's answer the question first. You're getting to a mechanism. Okay. Would we want it high iron, low iron? When would we want IRE-BP? We want it under all conditions because remember it's responding here to both low iron conditions and high iron conditions and has a role in both of those, right? So the answer to your question is we wouldn't want to have much regulation on it. We would want to be making it most of the time. Does that make sense? Okay. Excellent. Okay, good discussion as always. All right, so that finishes up what I want to say about regulation of gene expression. And I want to turn our attention now on the last thing that we'll talk about, or the next to last thing we'll talk about this term, sensory perception. And if you were, shall we say, frustrated at the lack of answers we have to some of the problems that we've addressed, you'll see it even more so in sensory detection of sensory detection. All right. This is a very active area of research right now. Okay, very active area of research. We probably know the least about touch, pain reception, those sorts of receptors. We know marginally more about smell, taste, and vision, but even there, our knowledge is very, very limited. Okay? I have a former student of mine who's presently at Harvard who is very actively studying smell perception, okay? Because we really don't understand a lot of components, not only of the reception, but also of the effects on behavior, which is very important for some animals. Okay. This figure briefly shows what we're talking about. Okay. So our senses, of course, come in mostly through our head. Touch, of course, is going to come in through anywhere on the body. smell through the nose, there's the eyes, there's taste, and there's hearing. And what you see in looking at these is you're not going to see complete pictures in most cases, but you're going to see some really beautiful and interesting systems for detecting things. Okay, so we've got systems for detecting things. That's really what we're talking about. I start with smell. Okay, smell is a sense that, of course, comes in through our nose, and it's a sense that other organisms sometimes have peripheral organs that help them to sense chemicals by a process like what we call smell. Okay? These chemicals, as you can see, are relatively simple, and if you look at them, you see these all have a pretty common feature about them, and that is that they're all fairly nonpolar, meaning that they can easily volatilize. Okay? So when we think of something that we smell, it has to be something that can get out into the air where we can detect it. Now, our sense of smell is remarkable in a couple of senses. Haha, no pun intended there. All right? Look at the, let's see, I think it's here. So here are some more things that we can sense, et cetera, et cetera. Nope, that's not what I wanted. I wanted to show you. Where was it? Here, okay, yeah, look at this guy, all right? Spearmint versus caraway, two very, very different things that we can smell, and their structures are identical. The only difference is, and I say identical, they're not quite identical, is one is a stereoisomer of the other. That's the only difference in those structures. They have exactly the same chemical formula. They have exactly the same chemical groups. One is the mirror image of the other one. Okay? What's that? Okay. That's remarkable. Okay? It's really remarkable to think that, well, what this reminds us, of course, is that what's doing the sensing is these guys are binding to proteins within our nerve cells of our nose. And proteins, you know, have a specific 3D structure. And as a consequence of having a specific 3D structure, they will bind to specific molecules that have a specific 3D structure themselves. What we sense with one, we will not sense with the other. Okay? Really cool stuff. Okay. Well, how does sense work? All right. Well, the nasal epithelium starts with what are called olfactory bulbs. And when we smell something, we basically have to get the smell into our nose and up to these olfactory bulbs. Right? Your nose is clogged. You don't smell well because that chemical isn't traveling very well to get up to your olfactory bulbs. It's in the olfactory bulbs that these protein receptors exist. These protein receptors, when they bind to a specific chemical that they recognize called an odorant, when they bind to an odorant, they initiate a nerve signal. And that nerve signal travels, okay, to a specific region of the brain and says, hey, I got something here. And that brain then says, okay, this is what it smells like. Okay? Or gives you that sense of what smell actually is. The signaling that happens during the process that I just described to you is shown right here. We see that the signaling is a G protein. It's a specific G protein called G olfactory. You can think of it as golf if you want to. That's an olfactory G protein. This is found within one of those olfactory bulbs that I just described to you. When the olfactory bulb recognizes an odorant, we get a structural change. And just like you've seen before, the structural change induces the G protein to bind, to drop GDP and replace it with GTP, as you see here. GTP bound to the G protein causes it to lose the beta and gamma units, just like you saw before. And it goes to adenylate cyclase, just like you saw before, which favors the synthesis of cyclic AMP. And cyclic AMP goes to a receptor, causes it to open and let in sodium and calcium, we created an ion gradient. Okay? That's how we detect an odorant that we pick up in our nose and carry to our olfactory bulb. That nerve signal then is carried back again to specific regions of the brain, and those specific regions of the brain say, ah, this one is firing, then therefore they've encountered this chemical. This is a smell. Okay? Makes sense? Yes?
But so this direction should be going as long as odorant is sitting on the goal, right? So how comes that we eventually it would just disappear?
So the odorant is sitting on the receptor. The gulf is inside here. The gulf is the G protein.
Okay.
So this is the receptor that's binding to this is the odorant that's binding to the receptor here.
Does the odorant make the reaction as long as the odorant is bound to the receptor? Does it make
As long as the odorant is bound to the receptor, it will stimulate this signal. That's correct. Yes?
Do we have a separate receptor for every chemical that we can sense?
Okay, so her question is, very good question, which is leading to my next point. Do we have a different receptor for every chemical that we can sense? Because we start thinking about, wow, man, there are a lot of things that I can smell. All right? Is that how I smell? The answer is no. We have a limited number of receptors. We don't necessarily know all of the receptors, but we have a limited number of receptors. Okay? And let me just get to that. Humans have about 30 active ones. Okay? As we look among mammals, okay, we see that humans, and in fact this is among primates, humans have the fewest number of receptors. What does that tell you? It tells you that over the evolutionary period that human beings have been evolving under, smell wasn't as important to us as it was out here. Being able to smell more different things may be important for finding food. It may be important for finding a mate. Okay? If we do the same thing and we include rats on this scheme, rats of course are not primates, but rats are mammals, rats will have on the order of 300 to 700. Okay? We don't... Yes? Okay, I'm getting there. So, yeah, good question. Back in the back. So how does the smell affect glycolysis and other things going on inside the cell? This is going on only inside of nerve cells, keep in mind. And so just like any nerve cell, when we have an ion gradient that's forming, it's only forming for a short period of time. So it's not going to have any significant effect on glycolysis. Okay? Yes?
You said that we have about 30 active ones. Does
Yes.
that mean we have evolutionary remnants?
Do we have evolutionary remnants? Do we have inactive receptors? And the answer is yes, we do. We have many that are not active, that don't function, that can't function because they've mutated to be non-functional. But we can see what they were. Yes? Okay? Now, what everybody wants to know, one more question, then I'll move ahead.
Can those inactive receptors can be activated by any chance? Because I know
Can the inactive receptors be activated? It's not a matter of that they just don't work. It's a matter of they've been mutated. They'd have to mutate back in order to function. So they're not functional as they are.
how come that some pregnant women they have very, very short chakras?
Let me not address that at the moment. Okay. I want to get to the question I think everybody wants to know is how do we get this pattern of smelling that we understand? Okay. The answer to that is here. What this shows is that it's a list of various odorants. Okay. These are various carboxylic acids that were given to people, and they mapped what receptor, first of all, bound to these odorants. And what we saw was that there wasn't exact things that were there. So contrary to what I told you earlier, all right, there was a pattern of binding. So here's various receptors 1 through 14. This C5 alcohol really only bound to this one with any affinity. The darker the binding, the darker the color, the more binding. Whereas the one that had 8 carbons bound to this one, it 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 bound to this one, et cetera, et cetera. This pattern of binding is what gives us the ability to distinguish different smells. Okay? So something that stimulates here, 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 here is stimulating those regions of the brain that correspond to those. And the brain says, well, this is different from C7 because, hey, I don't pick up that one over there. So therefore, it's a different smell. Now you start doing combinations of these and you begin to realize that, whoa, the number of smells that I can discern is enormous. Okay? It's absolutely enormous because not only is there a difference in which ones light up, but how strongly they light up as well. Okay. Let's see. What else is the other thing I want to say there? These, well, actually, I won't talk about that. I'll save that because I think that'll just raise other questions here. Okay, so that's what I want to say. Well, no, I like the questions, but they're not questions that I can answer for you here. So I'm not opposed to questions. Don't misread what I meant by that. I meant that I don't want to be able to raise things that I can't answer for you. What that was showing was how the bulbs converge. Okay, yes? Are there patterns that distinguish between pleasurable versus unpleasurable smells? Those things are done in your brain. And your brain is there to identify unpleasurable smells because it perceives them as dangerous to you. And it's designed to get you out of a situation. It may in some cases, it make you vomit. So that if you've eaten something that you're getting rid of it. Back in the back. It doesn't tell you anything about enantiomers. That's correct. Well, it could. I mean, so you have to keep in mind that there are only a few thousand, odorants that I could give. I'll get to my thing here. A few thousand of these that I could give, right? So I might have an enantiomer that binds, let's say, I don't know, this one down here, but not this one right here, right? They just have different patterns of binding. That's all that we're talking about. Okay? Yes? Is this dependent upon how many receptors we have? Is that what you said? Uh-huh. Okay, so basically the question is, what happens when we overload the system? It's the same when we overload any system. Any nerve signal is eventually going to be turned off. The brain's going to turn off response to that. And that's what's happening here when you overload on a smell. Yeah? How long do odorants stay bound to receptors? It depends. So it depends on the odorant. It's going to depend on the receptor, the affinity they have for each other. Things that don't bind with high affinity aren't going to be around as long as things that bind with higher affinity. They're not forming any bond because, remember, these are non-covalent interactions. Almost every interaction we see with a protein is a non-covalent interaction. Okay. So much for smell. Let's move to taste. Taste is actually a lot simpler than smell is in one sense. Okay? Taste, we really are only able to perceive about 5 different tastes, although recently people have claimed they might have detected a 6th one. They are sweet, salty, umami, bitter, and sour. Okay? You say, well, I can taste a heck of a lot more things than that. A steak tastes different than tofu, right? Well, our sense of taste is, to some extent, a blending of these. It's also, more importantly, colored by what we smell. Our sense of smell interferes with our sense of taste. Okay? So what we smell affects the perception of taste as well. So the sense of smell is a very strong sense in that regard. Yes?
The people who have poor sense of smell, where is their deficiency? Is it in the receptor?
Okay. So where is the person who has a poor sense of smell? I have a very poor sense of smell. Where is the deficiency? As you can imagine, it could be in a variety of places, but most commonly it probably is in the actual sensing olfactory bulb itself. Yes? What is umami? Umami is glutamate. It is a sort of a salty, meaty kind of a taste. I don't know how to describe it. It's kind of hard. How would I describe a taste? I mean, what's that? Savory. Savory. Okay. Okay. I knew senses would light everybody up. We sense taste through taste buds. And taste also uses a G protein in its scheme. I'm not going to show you the G protein, but I will give you the name of the G protein. And it's called Gustdusen. Gustdusen, yes. Well, it was on the screen just a moment ago. There it is right there. Gustdusen. It's a G protein, works very similar to what you saw before. Okay? Now, I should caution and say that not all of the 5 senses work via Gustdusen. We'll see one of them, salt, that doesn't use Gustdusen. It directly uses the salt as an ion channel, as the thing that opens the channel. And we'll see that in a second. Okay? So there's Gustdusen. You can see it lighting up. There it's shown in green here. It's shown in red over here. And when we look at the individual receptors that are in the membranes of the taste buds, what we see is that there's great conservation of them from one to another. They're very, very similar to each other. They don't differ an awful lot. On this figure, white shows the residues that are virtually identical across all of them, and red is pretty close. Okay? So only when we get to blue do we see much in the way of variation. And it's in those blues that allow the different senses, or the different tastes, to be perceived differently. Overall, the conservation of these is pretty good. Okay? If we look at a particular receptor, we can actually look at the signaling that a particular receptor does, okay, by the amount of GTP bound at the G protein. All right? If we feed these various chemicals to the receptor and see which one of them causes the G protein to get loaded with GTP. In this case, the only one that caused the G protein to get loaded was cyclohexamide, which is a very bitter compound. Okay? So that's the way in which we can test individual receptors and determine what they actually are binding to. Okay? Now, I'll show you a figure that's a little confusing. This contrasts the way that we perceive smell with the way that we perceive taste. And this actually tells us why we really don't pick up more than about 5 or 6 different tastes. Let's start with smell. Notice that with smell, I can go from a particular bulb with a particular receptor and map it to a specific region of the brain. Here's a different bulb with the same receptor shown in orange, and it maps to exactly the same section of the brain. The blues map to the same section, the grays map to the same section, etc., etc. When I go to taste bitter, I may actually have several different receptors, each of which can detect bitter, just like I had over here, but instead of being linked between bulb and brain, they're mixed. So my bitter receptor taste bud might have several different bitter receptors on it, and they all map to the same section of the brain. Therefore, I can perceive that it's bitter, but I can't tell what kind of bitter it is. I can't, for example, if I'm thinking about salt, I can't taste the difference between sodium chloride and potassium chloride, even though they're different chemicals, right? So this mixing that happens with the receptors and the lack of a specific region of the brain that each one links to is why our senses of taste are much more I want to say simple, but not as diverse as they are with respect to smell. Okay. Now, this is a very cool experiment I want to show you. So people wanted to understand, okay, what it was that we, and specifically we being, in this case, a rat, had its ability to taste something sweet. So they had some receptors, and they said, well, which one is responsible for this rat tasting or sensing sweet? All right? So what they did was they took mice, or rats, I forget if it was mice or rats, and they made mutations such that they had several groups of mice. they had receptors called T1R1, T1R3, and T1R2. And the question was, which of these receptors was responsible, which one was picking up the sweet taste for the rat? All right? So one rat had only the T1R1 receptor. And what they measured was they put this rat into a thing where they had, it could get a drop of a sugar water, and they counted how many times it would lick. When they sense sweet, they will go back and they will keep licking and licking and licking because they like sweet, and they would eat more. So the y-axis is plotting the relative lick rate. I like that, the RLR, right? The relative lick rate. And the relative lick rate, the more licks, the more they're sensing. Well, you can see that the one that had only the T1R1, one of the receptors, the rat didn't do hardly any licking. The one that had T1R2 did a little bit, but not much more. And the one that had T1R3 also did a little bit more, but not much more. But the only rat that had T1R2 with T1R2 with T1R3 licked like crazy. What that meant was that it was the combination of these two receptors that was necessary for the sense and the detection of sweet. People who work in sense labs really get to do some really cool experiments, and this is a pretty neat one that's there. Okay. Now, I said that salt was a little bit different, and salt, our ability to taste something salty, arises as a result not of a G protein, but of this receptor that you see right here in the taste bud of the salt receptors. Okay? This taste bud has a protein in its membrane that has an opening that will allow sodium to pass through it. When sodium is present, it causes this protein to change shape slightly, which opens it up, and sodium can flow in. Well, when sodium flows in, that's an ion gradient. Sodium is flowing into the cell, and what's happening is the cell is starting a signal. It's a nerve cell. It's going to send a signal, and the brain's going to pick it up. Okay? Now, if the sodium chloride isn't out here, then it goes back to its normal state. It closes up, and the signaling stops. Okay? So this is a really simple system. Okay? This system causes the signaling to happen as a result of the molecule itself being present. We know that because we can actually block that channel that I described to you before, and that the molecule that blocks the channel is a compound called amiloride. Amiloride, if you give it to I'm not sure you give it to a person or not because it might be poisonous, but if you give it to an organism and then you give them salt, they will be unable to detect salty taste. It completely blocks the salt signal. Because what it does, it goes in that protein and physically occupies it. So sodium can't go flowing through it. Okay. It's just a receptor protein. There's not a specific name, no.
Is it on nerve cell as well, right?
These are nerve cells, that's correct.
But within your cell probably also generates its own gradient, so the lots of sodium should be flowing outside due to the pump. So why is those just...
Yeah, so why is sodium flowing? Okay, so why is sodium flowing out or flowing in? Sodium could actually flow out or flow in, and either of those would cause a gradient change.
Yeah, but if you don't taste any sweet, you don't have any salt on our tongue, for example, but nerve cells still, they pump sodium out, and this sodium can come back. It flows next to it and can open this protein and flow inside, right?
I'm not sure I understand the question. So this protein is denatured by alterations in sodium concentration. Okay? So if I'm eating something salty, I'm giving it the ability to change its shape and open up so that sodium can move across the membrane. Right? Okay. So that's what's happening, and that's a very simple regulation. It's actually the simplest regulation we've seen of any of the senses so far. I hope I answered your question. Okay. All right. That's the sense of taste. Vision. Vision is pretty remarkable. I'm just going to get started on vision today. I'll finish vision on Friday, but vision is remarkable. Okay? The electromagnetic spectrum is shown here, and the part of the electromagnetic spectrum that we as humans can see is right there. A very tiny part of the electromagnetic spectrum. There are organisms that can see things that we can't see. They can see things in the infrared, which is more to the right. They can also see things further into the ultraviolet in some cases, which is a little bit to the left. Okay? But even those organisms don't see it very much further than we do. So their ability to sense electromagnetic radiation with vision is fairly limited compared to the spectrum of things that are out there. Our ability to sense light comes about as a result of this compound right here. This is a form of vitamin A. And this is why vitamin A is necessary for vision. Vitamin A looks like this guy right here. And you'll see it says 11 cis. If you look at the bonds out here, which as we number, this is numbered 1, 2, 3, 4, 5, 6. There's 7, 8, 9, 10, 11. We see a cis bond instead of a trans. There's a trans, a cis, a trans, and finally an aldehyde. Okay? The bond at position 11 is cis. Now, I've shown you before when I talked about bacteria rhodopsin the fact that vitamin A, in response to light, will change from cis to trans and back. From cis to trans and back. It's very, very sensitive to light. Very, very sensitive to light. Okay? If we look inside of rod cells give us the ability to detect light, not really to distinguish it much, but the ability to detect light. They look like this, kind of a cool figure that's there. And these rod cells have within them, these are the ends of nerve cells again, they have a, excuse me, they have a 7-TM in them. What's a 7-TM? Okay. 7-transmembrane domain, just like we saw with the beta-adrenergic receptor. Okay. They have a 7-TM, but the 7-TM that they have is a little different. There's the 7-TM you saw before. Here's a 7-TM that contains vitamin A. When light hits vitamin A, it goes from a, actually that's a little, what did they do there? Well, they've made another isomer in this, but it doesn't really matter for our purposes. You can see that the bond configuration of cis and trans has changed in going from here to here. And by the way, vitamin A will change all of its bonds as a result of action of light. The predominant one is changing the 11 from cis to trans. So cis to trans, remember the bacteria rhodopsin was kicking the protons out, was doing this? This guy, when it goes from cis to trans, is going to do what? It's going to change the shape of the 7-TM. And changing the shape of a membrane receptor, you know, is important in G protein signaling. It's that I'll talk about next time. But before then, I have a song for you. And the song actually relates to last lecture. It's called Chromatin. It's an old tune called Sunshine on My Shoulder. Anybody know that song? Okay. Side chains of the lysines bind to phosphates. Minus charges cling to pluses tight. Chromatin assembly is essential. Eukaryotic cells must get it right. Cells are tiny micro-scaled enclosures with nuclei tucked deep in their insides. That's the place, amazingly enough, that 7 feet of DNA resides. H2A and H2B have lysines. That's how they get charges, don't forget. Paired with H's 3 and 4, they make up a chromatin coarse octameric set. These get, it's the same melody all the way through. These get organized in higher orders, changing with the cycles of the cell. Denser packing going through mitosis. At other times the structures simply swell. So because of all the hyperpacking, nuclei can hold entire genomes. Thank the histone spooling DNA for physically downsizing chromosomes. Okay, see you guys on Friday.